SwayamPrabha. Digital India. Educated India.
Measurement of organic pollution. Lecture number 25. Let us now see, although we had seen how these analyses are done, some new analyses have been incorporated. So, let us go one by one and how the measuring of organic pollutants in the textile effluent is carried out in the laboratory. Let us try to understand them in more detail. Measuring organic pollutants in textile effluent is crucial for assessing its environmental impact and ensuring compliance with regulations. Several methods can be employed for this purpose. Chemical oxidation demand (COD) measures the amount of oxygen required to oxidize organic compounds in the effluent. It provides an overall indication of the organic pollution level. BOD, biochemical oxygen demand (BOD) measures the amount of dissolved oxygen consumed by microorganisms while decomposing organic matter in the effluent over a specific period of time, which is normally either 5 days, 7 days or 20 days. It reflects the biodegradability of organic compounds. So, COD and BOD are two major crucial parameters which need to be tested. Similarly, total organic carbon TOC measures the total amount of organic carbon present in the effluent. It provides a direct measure of organic content without the need of oxidation. Gas chromatography and mass spectrometry. This we had not discussed too much in detail. It is a new learning. GCMS is a powerful analytical technique used to identify and quantify individual organic compounds in the effluent. It offers detailed information about the composition of organic pollutants. High performance liquid chromatography HPLC. HPLC can also be used to separate and quantify organic compounds present in the effluent, particularly dyes and other complex organic substances. Fluorescence spectroscopy. This technique utilizes the fluorescence properties of organic compounds to detect and quantify them in the effluent. It can be particularly useful for monitoring aromatic and polyaromatic compounds. So, GCMS, HPLC, fluorescence spectroscopy, these are all very specialized techniques which are used for the analysis of measuring the organic pollutants in the textile effluent. Enzyme linked immunosorbent assay ELISA. ELISA kits are available for specific organic pollutants or classes of compounds. They provide rapid and sensitive detection, but they may be limited to certain target compounds. Microbial assay. Microbial assay such as respirometric method utilize the metabolic activities of microorganisms to assess the presence of organic pollutants in the effluent. They can offer a relatively quick assessment of biodegradability. When measuring organic pollution in textile effluent, it is important to choose appropriate methods based on the characteristics of the effluent and the specific pollutants of concern. Additionally, regular monitoring and analysis are essential to ensure compliance with environmental regulations and to implement effective pollution control measures. So, it is important to know how to analyze, what to analyze, and every time it should be kept in mind that the compliance factor should be adhering to according to what the environmental regulations have been laid down. The values should only remain in that stipulated value range. Measurement of COD. Although we had covered this in one of the lectures, we will re-look at it. Measurement of chemical oxygen demand in textile effluent is a common method to assess the level of organic pollution, and this is a well established fact by now. This is how COD measurement is typically conducted. Sample collection. Collect a representative sample of the textile effluent using appropriate sampling techniques to ensure accuracy and consistency. Digestion. The COD measurement typically involves oxidizing organic compounds in the sample. This is usually achieved through chemical digestion using a very strong oxidizing agent such as potassium dichromate in the presence of a catalyst, sulfuric acid. Titration. After digestion, the remaining oxidizing agent is titrated with a reducing agent, ferrous ammonium sulfate, to determine the amount of consumed oxidation agent during the oxidation process. Calculation. The COD of the sample is calculated based on the amount of oxidizing agent consumed during the reaction. The result is typically expressed in milligrams of oxygen per liter, milligram per liter of sample. Consideration. Digestion conditions. The digestion process must be carefully controlled to ensure complete oxidation of organic compounds without loss of volatile substances or interferences from inorganic compound. So, this has to be done very carefully. Calibration. Calibration of the apparatus and reagents is essential to ensure accurate measurements. Standard solutions with known COD concentrations are used for calibration purposes. Quality control. Regular quality control check should be performed to verify the accuracy and precision of COD measurements. We may just measure and we are not sure whether it is right or wrong. So, standards with known COD values have to be done for the calibration of the instrument, and periodically these analytical instruments have to be calibrated from time to time. This may include running duplicate samples and analyzing reference material, and sometimes even triplicates. Safety precaution. Handling of strong oxidizing agents and acids require appropriate safety measures to prevent accidents and ensure personal protection. So, all that good laboratory practices must be kept in mind. Equipment and reagent. COD reactor, a digestion apparatus equipped with heating and mixing capability, is used for the oxidation reaction. Titration setup. Equipment for titration including burettes, pipettes, and a suitable indicator ferroin is required for measuring the consumed oxidizing agent. Reagents such as potassium dichromate, sulfuric acid, ferrous ammonium sulfate are the primary reagents used in COD measurements. By following standardized procedures and ensuring proper quality control, COD measurements provide valuable information about the organic pollution level in the textile effluent, aiding in environmental monitoring and pollution control efforts. Let us now understand how measurement of BOD is done. We had studied this in the few lectures back, but we will re-look at it. Measuring BOD or biochemical oxygen demand in textile effluent is again a crucial assessment of the organic pollution level in wastewater. Here is a general outline of how it is done. Sample collection. Grab samples of textile effluent from different points in the discharge system to ensure representativeness. Storage. Store the sample in clean, airtight containers at a low temperature, usually 4 degrees, to minimize biological activity until testing. Initial dissolved oxygen measurement. Measure the initial dissolved oxygen level in the sample using a dissolved oxygen meter. This provides a baseline for comparison with subsequent readings. So, how much of the oxygen is already present? That much will be required for the biological degradation. Incubation. Incubate the sample in darkness at controlled temperature, usually 20 degrees, for a specific period of time, typically 5 days, but it could be 7 days or 20 days as well, depending on which BOD method we have adapted. During incubation, microorganisms in the sample will consume the organic matter, depleting the oxygen, dissolved oxygen. Final DO measurement. After the incubation period, measure the dissolved oxygen level again. The difference between the initial and the final DO readings indicate that the amount of oxygen consumed by microbial activity, which correlates with the BOD. Quality control. Perform quality control check to ensure the accuracy and reliability of the test results. This may include running duplicate or triplicate blanks and standards. Reporting. Report the BOD value along with any relevant information about the sample collection, analysis condition and quality control measures. It is essential to follow standardized methods such as those outlined by organizations like American Public Health Association, APHA, or International Organization for Standard ISO, to ensure consistency and comparability of results. Additionally, regulatory agencies may have specific guidelines to limit for BOD levels in textile effluent that must adhere to. Measuring TOC. This we had not done in detail, so let us look at it how measurement of TOC is done, which is total organic carbon in textile effluent. It is another important parameter for assessing the organic pollution content. Here is a general procedure for measuring TOC. Sample collection. Collect representative samples of the textile effluent from various points in the discharge system. Storage. Store the samples in clean, airtight containers at a low temperature, typically at 4 degrees centigrade, to prevent microbial activity and preserve the total organic content until testing is done. Preparation. If necessary, filter the sample to remove suspended solids that could interfere with the TOC analysis. dilute the sample if the organic carbon concentration is too high for accurate measurements, because it is of general understanding that the more dilute the solution, the more accurate is the result, because concentration can cause turbidity and interference in the analytical process. Instrumentation: use a TOC analyzer to measure the concentration of organic carbon in the samples. TOC analyzers employ different methods such as combustion oxidation or UV persulfate oxidation, followed by detection of resulting carbon dioxide. So, somehow either by combustion oxidation or UV persulfate oxidation, the oxygen content is evaluated in terms of the release of carbon dioxide. Calibration: calibration of TOC analyzer is done by using standard solution of known organic carbon concentration to ensure accurate measurements. Analysis: run the samples through TOC analyzer according to the manufacturer's instructions. The analyzer will oxidize the organic carbon present in the sample and measure the resulting carbon dioxide, from which the TOC concentration is calculated. Quality control: perform regular quality control checks using certified reference materials to verify the accuracy and precision of the TOC analyzer. So, as I told that each of these equipments have to be calibrated time and again periodically, so that they give precise result. Measuring TOC: reporting: report the TOC concentration in the textile effluent along with relevant information about sample collection, preparation and analysis condition. Monitoring TOC levels in textile effluent is essential for assessing the overall organic pollution load and ensuring compliance with regulatory standards. It is also useful for evaluating the effectiveness of wastewater treatment processes and identifying opportunities for improvement in pollution prevention and control measures. Measuring organic pollution in textile effluent through GCMS. This analytical method we had not dealt in detail. Using gas chromatography mass spectrometry, that is GCMS, to measure organic pollution in textile effluent is a powerful analytical technique, and this is how it works. Sample preparation: first, the effluent sample needs to be prepared for analysis. This involves filtration to remove particulate matter and potentially concentrate the organic compounds of interest. So, in GCMS, there is a very specialized step of sample preparation. The sample cannot be just taken like this and analyzed, unlike what we do in COD and BOD and TOC. Gas chromatography: in GC, the sample is vaporized and injected into chromatograph. The GC separates the mixture into its individual components based on their different affinities for a stationary phase. Typically, a thin layer of liquid or polymer and a mobile phase, an inert gas like helium. The compounds travel through the column at different rates, separating them based on their size, polarity and other chemical properties. So, there is a solid stationary phase, which is the wall of the GC column, and there is a mobile phase, which is helium gas, which is carrying. Now, Due to adsorption, desorption, adsorption, desorption, the compounds start moving and they separate, and according to the polarity and their affinity for the coating material, that is the solid phase, they get separated. Mass spectrometry. As the separated compound exit the GC column, they enter the mass spectrometer. Here they are ionized typically by electron impact, which fragments them into smaller ions. These ions are then accelerated through an electrical field and into a mass analyzer, which separates them based on their mass to charge ratio. The resulting mass spectrum provides information about the identity and quantity of the compound present in the sample. Identification and quantification. The mass spectra obtained from the GCMS analysis can be prepared and compared to database of known spectra to identify the organic pollutant present in the textile effluent. They have big libraries. One of them is called NIST library. So, from the library search one can identify the peak that has appeared in the spectrum. Additionally, the peak area and heights of the chromatogram can then be quantified for concentration of each compound. Data analysis. Now, this is very crucial for interpretation of the mass spectrum. GCMS. Once the compound are identified and quantified, statistically analysis and interpretation can be performed to assess the level of organic pollution in the effluent. This may involve comparing the results to regulatory standards or previous measurements to determine the environmental impact and potential risks associated with the pollution. So, either it can be compared with other known compound or it can be searched through library because of the mass fragmentation pattern matching. Reporting. Finally, the results of the analysis are typically reported in a comprehensive manner, including details of the compounds detected, their concentrations, any relevant regulatory limits and recommendation for remediation or further action if necessary. GCMS is a widely used technique in environmental analysis due to its sensitivity, selectivity and ability to identify a wide range of organic compounds present in complex samples like textile effluent. How organic pollutants are analyzed on HPLC? We just now saw how they were analyzed on GCMS. Measuring organic pollutant in textile effluent through high performance liquid chromatography, that is HPLC, is another common analytical method, and this is how it is done. Sample preparation. As with GCMS, sample preparation is crucial here also. Textile effluent samples need to be filtered and possibly concentrated to remove particulates and ensure that organic compounds of interest are present in the suitable concentrated material which will be used for analysis. High performance liquid chromatography. In HPLC, the sample is injected into a column packed with stationary phase, usually a porous material like silica, and pumped through the column at high pressure. The mobile phase, that is the solvent, carries the sample through the column. As the sample moves through the column, compounds in the sample interact differently with the stationary phase based on their chemical properties, causing them to separate. Here also, just like GC, there is absorption desorption on the solid surface, and here of course it is not gas helium which is the carrier gas, but here it is solvent which is carrying the compounds, and during their passage over the column they get separated due to their nature, inherent nature of their polarity. Detection. After separation, the compounds elute from the column and enter a detector. Common detectors used in HPLC for organic pollution analysis include UV visible spectrophotometer, fluorescence detector, and mass detectors. UV visible detector are often used for compounds that absorb UV or visible light, while fluorescence detectors are sensitive to fluorescent compounds only. Mass spectrometer can provide additional structural information about the pollutant molecule. Identification and quantification. The retention times of the compounds in the chromatogram can be compared to standards or databases to identify the organic pollutants present in the textile effluent. Additionally, the peak areas or heights in the chromatogram can be used to quantify concentration of each compound. Data analysis, just like what we did in GCMS, and we saw here also the results obtained from HPLC analysis can be statistically analyzed and interpreted to assess the level of organic pollution in the effluent. Comparisons to regulatory standards or previous measurements can help determine the environmental impact and potential risk associated with these pollutants. Reporting. Finally, the results of the analysis are typically reported comprehensively, including details of compounds detected, their concentrations, any relevant regulatory limits, and recommendation for remediation or further action if necessary. HPLC is advantageous for its ability to separate a wide range of compounds, including polar and non-polar compounds, making it suitable for analyzing complex mixtures like textile effluent. Additionally, HPLC can often provide faster analysis times as compared to GCMS. However, it may not offer the same level of sensitivity or compound identification capabilities as GCMS. So, each one has its own advantages. Now, let us see how fluorescence spectroscopy works. Measuring organic pollutants in textile effluent through fluorescence spectroscopy is a rapid and sensitive technique that exploits the intrinsic fluorescence properties of the organic compounds which are present in the textile effluent, if at all they are. Here is how it typically works. Sample preparation. Textile effluent samples are usually filtered to remove particulates and dilute to an appropriate concentration to ensure accurate measurements. Some pre-treatment steps might be necessary to remove interfering substances or enhance the fluorescence signal of the target compound. Fluorescence spectroscopy. The prepared sample is then analyzed using a fluorescence spectrophotometer. This instrument emits light of a specific wavelength, that is the excitation wavelength, onto the sample, causing the organic compound within the sample to absorb this light and become excited. As the excited molecule return to their ground state, they emit light at longer wavelengths, a phenomena which is known as fluorescence. Detection. A detector in the fluorescence spectrophotometer measures the intensity of the emitted fluorescence at specific wavelength. Different organic compounds exhibit characteristic fluorescence spectra. So, two compounds will not have similar spectra, and that is the advantage, allowing for identification and quantification of various pollutants present in the textile effluent. Calibration. To quantify the concentration of organic pollutants, calibration curves are typically constructed using known standards with known concentrations. These calibration curves relate the intensity of fluorescence emission to the concentration of the target compounds, allowing for the determination of unknown concentrations in the sample. Data analysis. Once the fluorescence spectra are obtained, data analysis involves comparing the emission spectra of the sample to those of the known standards or databases to identify the organic pollutant present. Additionally, the fluorescence intensity can be correlated with the pollutant's concentration using a calibration curve. So, it is quite similar to that of UV, where Beers-Lambert's law follow. So, whatever is present will be reflected in terms of its potential to emit the light. Reporting. The results of analysis are reported including identity and concentration of the organic pollutant detected in textile effluent. This information can be used to assess the level of pollution and compliance with regulatory standards as well as to guide remediation efforts if necessary. Fluorescence spectroscopy therefore offers several advantages for measuring organic pollution in textile effluent, including high sensitivity, rapid analysis times, and the ability to detect a wide range of organic compounds. However, it may require careful calibration and validation to account for interferences and matrix effect from complex samples like textile effluent, which means that interferences and matrix effects have to be corrected. Then let us see how ELISA works, how it helps in measuring the organic pollutants in textile effluent. Measuring organic pollution in textile effluent through enzyme linked immunosorbent assay, ELISA, involves utilizing antibodies to detect specific organic compounds, and this is how it works. This is also a new learning. Antibody selection. Specific antibodies that bind to target organic pollutants present in textile effluent are selected or developed. These antibodies should have high affinity and specificity for the target compounds to ensure accurate detection. Sample preparation. Textile effluent samples are prepared by filtering to remove particulates and potentially dilute to an appropriate concentration for analysis. Pre-treatment steps might be necessary to remove interfering substances or enhance the detection of the target compounds. Immunoassay procedure. The prepared sample is then added to a microplate coated with selected antibodies. If the target organic pollutant are present in the sample, they will bind to the antibodies immobilized on the plate. Detection. After allowing time for binding to occur, the microplate is washed to remove any unbound substance. a secondary antibody linked to an enzyme such as horseradish peroxidase or alkaline phosphatase is then added. This secondary or antibody binds to the target compound antibody complex. Enzymatic reaction. A substance specific to enzyme linked to the secondary antibody is added. If the target compound is present and bound to the antibodies of the microplate, the enzyme catalyzes a reaction with substrate resulting in the production of detectable signal, color change or fluorescent signal. So, that is how it works. Signal measurements. The intensity of the signal generated is proportional to the concentration of the target organic pollutants in the sample. This signal can be measured using spectrophotometer for calorimetric assay or for fluorescence reader for fluorescence assay. Data analysis. The measured signal is compared to a standard curve generated using known concentration of target compounds. This allows for the quantification of the target pollutant in the textile effluent sample. Reporting. The results of the ELISA analysis, including the concentration of organic pollutants detected in the textile effluent, are reported. This information can be used to assess the level of pollution and compliance with the regulatory standards, as well as to guide remediation efforts if necessary. ELISA offers several advantages for measuring organic pollution in the textile effluent, including high specificity for target compounds, ease of use, and relatively rapid analysis times. However, it requires prior knowledge of the target compound and the availability of specific antibodies for detection. So, it requires a concerted effort to match the antibody with the target pollutant. Additionally, ELISA may not be suitable for detecting a wide range of organic pollutants to other analytical techniques like chromatography or spectroscopy. Now, let us see how microbial assays are carried out for measuring organic pollution, and this is also new learning. Measuring organic pollution in textile effluent through microbial assay involves using microorganisms as indicators of pollution level, and this is how it works. Selection of microorganism. Specific microorganisms or microbial communities are selected or developed as indicated for organic pollution. These microorganisms may include bacteria, fungi, algae that are known to respond to the presence of organic compounds in the environment. So, there is a development stage of indicator. Sample preparation. Textile effluent samples are prepared for microbial assay by filtering to remove particulates and adjusting the pH and nutrient levels if necessary to create suitable condition for microbial growth. Inoculation. The prepared sample is then inoculated and with the selected microorganism. This can be done by adding a known quantity of cultured microorganism or by directly inoculating the sample with environmental samples containing indigenous microorganism. So, both way it can be done. Microorganisms that are present in the effluent or specialized microorganisms which have been identified. Incubation. The inoculated samples are incubated under controlled conditions, that is temperature, pH and oxygen level. for a specified period to allow the microorganisms to grow and respond to the organic pollutant present in the effluent. Monitoring microbial growth or activity. Microbial growth or activity is monitored over time using various methods. This may include measuring changes in cell density, metabolic activities, or the production of specific biomolecules, that is the enzymes, associated with pollutant degradation. Data analysis. Data obtained from microbial assay are analyzed to assess the level of organic pollution in the textile effluent. This may involve comparing microbial growth or activity in the sample to control samples or reference standards to determine the extent of pollution. So, always there will be a reference or a control sample against which what is the microbial assay result of the pollution or the pollutant. Reporting. The results of the microbial assay include any change in microbial growth or activity associated with organic pollution are reported. This information can be used to assess the environmental impact of the textile effluent and guide remediation efforts if necessary. Microbial assay offer several advantages for measuring organic pollutants in textile effluent, including their ability to provide real-time information on pollutant degradation processes, their cost effectiveness, and their applicability to a wide range of organic pollutants. However, microbial assay may be influenced by factors such as the composition of the microbial community, environmental conditions, and potential interference from other substances in the sample. Additionally, microbial assay may not provide detailed information on the specific organic compounds present in the effluent compared to other analytical techniques like chromatography and spectroscopy. So, each method has its own importance. We cannot say that chromatography is the best or spectroscopy is the best or microbial assay is the best or ELISA is the best. Each one can do one specific job in a very good manner. There are many benefits associated if we measure the pollutants in the textile, because only then we can strategize the technology that would be required for the remediation. Measuring organic pollution in the textile effluent offers several benefits. Environmental protection, the first and the foremost. Textile effluent can contain a variety of organic pollutants including dyes, solvents, sizes and other chemicals used in the textile production. Measuring organic pollution helps identify and quantify these pollutants, allowing for better management and mitigation of their environmental impact. By understanding the extent of pollution, appropriate measures can be taken to protect aquatic ecosystem, soil quality and overall environmental health. Regulatory compliance. Many countries have regulations and standards in place to limit the discharge of pollutants into water bodies. Measuring organic pollution in textile effluent helps industries comply with these regulations by ensuring that pollutant levels are within acceptable limits. Regular monitoring and reporting of pollution levels also demonstrate a commitment to environmental responsibility and sustainability. So, regulatory norms have been set. and many countries have regulations and standards in place where limits of discharge of the pollutants are already stipulated, and we have to responsibly adhere to those values and abide by the regulation. Public health protection. Organic pollutants in textile effluent can pose risk to public health through contamination of drinking water, resources of exposure to contaminated soil and sediment. Measuring pollution level helps identify potential health hazards and allows authorities to implement measures to protect public health such as water treatment or contaminated site remediation. Reserve and resource conservation. Textile production consumes significant amount of water and energy, and the discharge of the polluted effluent can further strain natural resources. By measuring organic pollution and implementing pollution prevention measures, textile manufacturers can reduce their environmental footprint and promote resource conservation. This includes implementing cleaner production techniques, recycling water, and reducing the use of hazardous chemicals. Cost saving. Pollution prevention and control measures such as optimizing production processes and implementing wastewater treatment technologies can lead to cost saving for textile manufacturers in the long run. By measuring pollution level and identifying opportunities for improvement, companies can reduce waste generation, minimize regulatory fines, and improve overall operational efficiency and reduce the environmental impact. Sustainable practices. Measuring organic pollution in textile effluent is part of a broader commitment to sustainable business practices. By reducing pollution level and minimizing environmental impact, textile manufacturers can enhance their reputation, attract environmentally conscious consumers, and contribute to a more sustainable future for the industry. Overall, measuring organic pollution in textile effluent is essential for protecting the environment. That is the first and the foremost idea, public health and natural resources, while also ensuring regulatory compliance and promoting sustainable business practices within the textile industry. Challenges that one faces while measuring organic pollutants. Complexity of effluent composition. Textile effluent is a complex mixture containing various organic compounds including dyes, sizes, solvents, surfactants, and other chemicals used in textile manufacturing. This complexity makes it challenging to identify and quantify individual pollutants accurately, especially when multiple compounds are present at different concentration levels. Diverse range of pollutants. Textile manufacturing involves the use of wide range of chemicals, each with its own properties and behaviors in the environment. Some pollutants may be readily biodegradable, while others may persist in the environment for extended periods. Measuring such a diverse range of pollutant requires the use of multiple analytical techniques and approaches, adding complexity to the analysis. Low concentration. Some organic pollutants in textile effluent may be present at low concentrations, making them challenging to detect and quantify accurately. This is particularly true for pollutants that are highly dilute and need to be quantified accurately. So, diluted solutions of the effluent for those that degrade rapidly upon discharge are main problems for analysis and challenges. Achieving sufficient sensitivity in analytical methods to detect low concentration of pollutants can be technically challenging. Interferences and matrix effect. Textile effluent often contains a high level of organic and inorganic constituents which can interfere with each other, and analytical measurements can be affected that would affect the performance of the detection method. Matrix effect such as change in pH, salinity, turbidity can impact the accuracy and precision of analytical results, requiring careful sample preparation and method validation. So, there are challenges, although it is recommended, many methods are very apt for analysis, but still they are very challenging issues, especially for low concentration pollutants, for matrix interferences and so on. Availability of reference standard. Obtaining reference standard for all the organic pollutants present in textile effluent can be difficult for an analytical lab, particularly for emerging contaminants or compounds with limited commercial availability. Lack of suitable standards can hinder accurate identification and quantification of pollutants, leading to uncertainties in the measurement results. Analytical techniques selection. Choosing the most appropriate analytical technique for measuring organic pollutant in textile effluent depends on factors such as type and concentration of pollutants, sample matrix characteristic and available resources. Selecting the right technique requires expertise and careful consideration of advantages and limitations of each method. So, it is not an easy job. Analyzing is a textile effluent which is having an array of compounds. Which method would be most appropriate and suitable is pretty challenging. Regulatory compliance. Meeting regulatory requirements for pollutants discharge limit and reporting can pose challenges for textile manufacturers. Ensuring accurate and reliable measurement of organic pollutants in effluent is essential for demonstrating compliance with environmental regulation and avoiding potential fines and penalties. Addressing these challenges require collaboration among the researcher, regulatory agencies and industry stakeholders to develop robust analytical methods, improve pollutant detection technologies and promote pollution prevention and control strategies within the textile manufacturing sector. Now, it is for us to understand what is important and which pollutant should be analyzed in which manner, and therefore, it is mandatory to understand for an analyst how it should be done, why it should be done, how accurately this method can be followed. So, there is a great need for development of robust analytical methods. Improving the pollutant detection technologies and promote pollution prevention and control strategies. So, if we can reduce the load, our job becomes easy, and that is why we dedicated one lecture on pollution control. So, to conclude, in conclusion, measurement or measuring organic pollutants in textile effluent is crucial for protecting the environment, public health and natural resource undoubtedly. This is the focus. Despite the challenges involved such as complexity of effluent composition, diverse range of pollutants and technical limitations of analytical techniques, efforts to accurately quantify and monitor pollution levels are essential for sustainable textile manufacturing practices. Measuring organic pollutants in textile effluent is essential for achieving environmental sustainability, regulatory compliance, public health protection, resource conservation and the promotion of sustainable business practices within the textile manufacturing sector. So, it is a big responsibility, it is not a small responsibility. We have to look into how environmentally it can be made sustainable, how this textile effluent business can be controlled from making negative impact on the environment, how to follow the regulatory compliances, how to protect the public health, how to conserve the reserves and resources, and how to promote the sustainable business practices which is the need of the hour. By addressing the challenges associated with pollution measurement and adopting proactive pollution prevention strategies, stakeholders can work together to mitigate the environmental impact of textile production and ensure a cleaner and healthier environment for future generation. Therefore, what it boils down to that all the practices are very important, because pollution will happen. If textile has to be produced, there will be textile effluent generated in any case. But to measure, to recover and to analyze, these are three different parameters. First, we need to measure how much of the pollutant is still present in the textile effluent which is ready for discharge. It cannot be discharged without taking proper measures of mitigating or removing or degrading the organic pollutants. Once it is safe to discharge, then it can be done. So, we have understood now in this lecture that measuring all these parameters with one or the other, whether we use HPLC, whether we use GCMS or whether we use ELISA test or microbial assay. The bottom line is that we should have the true picture of what is the actual situation of the textile effluent pollution load. Having understood that, then we can be assured that we can make mitigation studies. So, with this we have come to an end of this lecture. Thank you.